All right. So, well, hey, good afternoon, everyone. Today we had the nice and big pleasure to have Dr. Cheri Kubota. She is a professor in the Department of Horticulture and Crop Science and director of the Ohio Control Environment Agriculture Center at the Ohio State University, at the Ohio State University. Dr. Kubota received a PhD in horticultural engineering and master's in horticultural science from Chiba University in Japan. She worked as a faculty for 6 years in Chiba University and then moved to Arizona, worked 16 years in the School of Plant Sciences at the University of Arizona. Dr. Kubota joined the faculty at Ohio State University just not too many years ago, 2017. So currently she is the research division vice president and also a fellow of the American Society for Horticultural Science. With that, Cheri, again, thank you. And I will mute myself. And then please, all the participants, just pop up questions in the chat. And then between Trish and myself, we'll monitor that later on. And hopefully will be time for questions.
Great. Well, thank you so much. Thank you, everyone, being with me today and listening to my talk. This is such a great series of seminar. I'm so glad I'm part of it. So I have been working on controlled environment agriculture, and pretty much what I do is a technology development, science-based technology development to advance controlled environment agriculture in the US and beyond. So what I do is not the basic science type of work. I don't do discovery work, but I take someone's discovery or understanding of science into innovative applications. So that's what I do. So that's why I don't limit myself to specific crop species or specific stage of the crop growth. I work on fruiting vegetables, small fruits, seedlings, young seedlings of vegetables, or leafy greens, and sometimes ornamental crop also, but very, very small portion, mainly working on food crop. So I'm not sure if you're already familiar with this because you are listening to all the controlled environment talk, but US is relatively behind compared to other countries in terms of usage of controlled environment in food crop production. US is very strong and advanced in terms of greenhouse ornamental floriculture crop species or ornamental crop production. So if you look at the statistics, so this is area under some kind of protection, greenhouse included. 90% or so are basically floriculture ornamental species, and then only 10% or so are food crop. When I came to US 2002 as a faculty member in University of Arizona, probably it was not even shown in this type of statistics. I'm talking about food crop, but now we have 10%. So that's a big jump. And then if you look at the breakdown of the food crop going into greenhouse or other type of protection, majority is tomato. And then when I started in Arizona, it was somewhere like 70%. So the increase was pretty much coming from, well, tomato increased also, but other crops like leafy greens, lettuce, herbs, those are actually the fast growing crops. So why I moved to Ohio, there are many reasons, including personal reason, but Ohio is one of the fast growing state in terms of using greenhouses or other controlled environment systems for growing food. So there's statistics from USDA showing that ratio between year 2017 to 2012 regarding the food production greenhouse area. And then I put all the states, including Texas, well known as leading states as using controlled environment for food production. Compared to those, I was quite happy to see Ohio big increase, almost 300% comparing 2 years of sample. And then that is pretty much driven by the newly introduced Canadian companies who are doing greenhouse tomato production. So the same period of 5 or 6 years, you see that more than 500 times increase over that. So if you are familiar with controlled environment agriculture or greenhouse production, you know that there are many areas of expertise involved. Just listed up the key expertise areas. My lab pretty much functioning in those 2 areas: plant science, plant physiology, whole plant physiology, and then also engineering, some part of engineering, particularly energy and mass balance is my area. And then also we collaborate with many other scientists and engineers, economists to do important projects to advance, again, controlled environment agriculture. So as I said, I work many different projects, including something like a most advanced artificial intelligence type of work, or more conventional horticulture, plant, physiology type of work. And then instead of doing a presentation showing all kinds of work, I decided to today just focus on something going on in the lab or the relevant topics today in this presentation. So right now, my lab is working on mainly 3 areas. One is vegetable grafting. And vegetable grafting is quite unique. It's a high quality transplant production technique. And then I see that area as a sort of interface between controlled environment and conventional open field crop production. So bridging those 2 technologies together to make the whole system more sustainable. The second topic I want to share a little bit is a greenhouse strawberry, which is a new, relatively new crop in North America in the greenhouse. So I like to talk about that. The third area is a hydroponics leafy green production. I'm working with plant pathologists on a low-cost disease management technique, and then also TIPAN, which is an environmental disorder, which is a kind of long-term program of controlled environment. So those things I'm gonna introduce today. Okay, so vegetable grafting. So it's an old and new technology, old because it was started probably around 1920s, new because the US hasn't fully, introduced that technology yet. Greenhouse growers started using that probably late 90s. And then those are the hydroponic greenhouse growers growing tomato year-round base. But most recently, watermelon is also, a small portion of watermelon field is now grafted. It's still very small, but I think this is going to increase. Why grafting? Two plants together like this one, you have to achieve disease resistance. And then also you expect yield increase, which is the main driver of using grafting in greenhouse tomato production. And then another benefit expected is a high quality fruit production because of the grafting. So one of the major achievements and then contributions my lab made was developing technologies for indoor transplant production. Actually, this is one of my long-term projects. Started back in Chiba University, Japan. We were working on indoor transplant production technology development using this multi-layer system. So this is more like a vertical farm setting, but instead of food crop to eat, those are seedlings to be planted in greenhouse or planted in the open field. And then because of the high density production, it makes sense to put electricity, high cost, because power plant cost is very, very small, sometimes less than 1 cent. So that's why we did that technology, and then became a sort of one of the standard technologies nurseries use for grafted plant production. And then also I contributed to introducing that technology in US, and then one company started several years ago using this technology. At the beginning, the lighting technology of those indoor transplant production was mostly fluorescent lamps because LEDs were very expensive. But as soon as nursery growers who use indoor transplant production approach started using LEDs, more advanced lighting technology, they started seeing issue. The issue is intumescence. That's a physiological disorder, particularly in tomato seedlings. And then you can see that gold-like, structure. It's almost like a, cancer look. But anyway, those are basically killing plants. And then the most widely used rootstocks cultivar in North American and European countries is the most sensitive. So every time you want to grow that rootstock, you have this issue of intumescence. We know that causal factors. Number one is the UVB deficient light environment. So that when we were using fluorescent lamps usually emit very small amount of UVB, and that was enough to prevent this issue. So all the tomato plants, regardless of the cultivar rootstock, could grow really healthy under fluorescent lamps. But as soon as switching to LEDs, you start seeing this intumescence issue because of the UVB. Another thing we know, research was done in University of Wisconsin back in late 1980s. The discovery was that intumescence induction was phytochrome related. So the phytochrome was involved in the induction process. So red-rich light environment is basically creating active phytochrome and therefore more susceptible for the intumescence. So one of our research earlier was showing the UVB, was basically confirming UVB is the real issue, or one of the real issues under LEDs, because LED doesn't emit UVB. So without UVB supplementation, this is one of the interspecific tomato rootstock, Buford, just looks so miserable. They are unhappy, unhealthy. They don't want to live. They want to die. But as soon as you start adding UVB as a supplemental lighting, you can see that the dose we provided by 6 hours of lighting during the nighttime we did, you can see that almost normal healthy plant growth. So it was quite interesting, or it's actually known, but actually first time I think we identified the minimum dose required for UVB supplementation to make the plants healthy. But this dose is actually only 1/10 of the dose you might get outdoor during midsummer day. So the dose itself is not that too high. However, if you consider this application in a nursery setting, people are working, you probably don't want to put UVB. You may have some issue of putting UVB in a nursery setting. So we wanted to develop something else or a different approach also. So what we did was approach based on the light quality or lighting technology. So one of the early research we did, my postdoc or former graduate student, Ricardo Hernandez, did was testing different ratio of red and blue to see what light quality is the best for growing tomato seedlings, scion and rootstock used for grafted plants. And what he found was, in addition to growth, intumescence incidence was different for different light qualities. So fluorescent light, of course, because of UVB, there was no incidence, so the zero incidence of intumescence. Unique if plants are grown under 100% blue light, no incidence of intumescence either. But plants are stretched and they're not really high quality. So this is good, but this is not applicable. Red and blue mixed LEDs having always high incidence of intumescence. And then if you increase the blue percentage or blue photon flux, then you see the trend of declining incidence. So that was interesting. But it's not really acceptable level. If 50%, incidence is still very high for tomato plants. So we did another approach based on the understanding developed in late 80s that phytochrome is involved. So we wanted to play with far red. And I know we could do during the day just supplemental far red to change the phytochrome equilibrium. But to do that, because of the red light, you have to apply a lot of far red light to cancel that red effect. So what we did was end of day approach. End of day light quality application is quite unique. You only need a teeny tiny amount, just enough to convert the phytochrome. So we started, different dose changed by time and intensity of far red at the end of the day or beginning of the night. So no other lights exist. And then we found that teeny tiny amount, far red photon, about 5 micromoles, 3 minutes or so, enough to reduce the intumescence. So it was quite unique, but plants stretch again because it's the end of day far red. So it's not really ideal either. So we decided to combine those two approaches, but because blue light can make the plants more compact. So if we can combine high blue, like 50%, 60% blue percentage with the end of day far red, we thought it might actually work. And then actually it did. So the next slide is showing, this is a 10% blue, 90% red. So the most, highest intumescence severity in this case, and then reduced by far red at the end of the day, or blue alone also reduced the severity of intumescence. If we combine those two, significantly reduce the intumescence incidence and then also severity. So you can see that plants looking more normal. This is basically the problematic condition, far red and blue. So it was quite unique. So we always recommend this light quality for growing plants now, particularly sensitive tomato under LEDs. And then now, tomato plants, some growers actually not just seedlings, but mature crop. Like there's one company doing that in Ohio also, and then they have an intumescence issue. So they are selecting cultivars, but if they want to have a cultivar and sensitive to intumescence, maybe light quality approach like that could help. So as I said before, grafting is new, relatively new. When I started in Arizona, there was no grafting capacity in the US. So most of the grafted plants, almost all the grafted plants came from Canada because Canada was more advanced in terms of technology for greenhouse. So grafting nurseries were there to support greenhouse tomato growers. But recently we did a survey after working many years for grafting technologies and then realized that it's quite a bit of grafted plants are now produced in North America, including US. And the US has several grafting nurseries like that, having a sizable capacity of grafted plants. The total number of grafted plants is about 60 million plants. And then 2002, when I started, it's probably 90 or more than 90% tomato, but now 24% watermelon, and then some small percentage for other crops. So it's quite a bit of technology advancement in terms of nursery technologies for vegetable seedlings. But still, number of grafted plant or grafting nurseries is very limited. So that means plants are traveling a lot, long distance from East Coast to West Coast, West Coast to East Coast. If you are familiar with vegetable seedling production, usually nurseries are located nearby the production site, so the local supply is available. But because of the transitional time of grafted seedling production technology, plants are traveling a lot, thousands of miles. So there are cases like grafted plants produced in a warm, no, the other way, cold area, like North Carolina or Canada in the middle of the winter. And then plants need to travel and then reach the warm area like Florida, Southern California, Southern Arizona. So in that case, the question comes, what kind of temperature they can tolerate? Do we know? We know some, right? The low temperature tolerance of different crops, but acute stress, we don't know much. So we were interested in finding out how or what acute stress of low temperature would cause plant growth and development after transplanting. So just show you a little bit of example here, what's going on in a shipment or during the shipment of grafted plants. So this is a just short period of time, 2-day transportation. Plants can travel all the way to, I guess, 5 days or so in North America, but this is a short shipment, example shipment from North Carolina to Ohio in the middle of the winter time. So that they were loaded into the container or pallet in North Carolina in a nursery, so the nice temperature around 20 degrees C or 68 degrees Fahrenheit, and then started declining temperature, and then reaching to the lowest 3.2 degrees C in Columbus, Ohio, when we opened that, basically the box of seedlings. We know that tomato plants can tolerate up to 10 degrees C. If you ever intend to either store the plants or grow the plants in whatever the environment, below that you want to avoid the chilling stress. But acute stress like that, how many hours plants can tolerate, we don't know. So that's, again, the sort of beginning of this research. So instead of working on tomato, we decided to work on watermelon because watermelon is one of the grafted crops in North America, and then also very widely grafted worldwide. And then also the most sensitive to chilling stress. Usually you don't want to go below 15 degrees C or 12 degrees C for watermelon. But again, during the shipping, you may have 1 degree C or even freezing temperature, which is not ideal, but during that 4 or 5 days of shipping. So my graduate student, John Otto, started working on this topic, and then several things he found. Freezing point of watermelon plant we tested was between -3 to -2 degrees C, so you don't want to go below that at all. And then we confirmed that chilling response was cumulative. So it's basically the product of temperature and also duration of exposure. So he set up a bunch of different combinations of durations and temperatures. Temperature was, I believe, somewhere from -1 degree C, so that's just above the freezing point, all the way to 3 degrees C. Combining hours of exposures up to 48 hours, I think maximum duration. And they wanted to see any correlation or any, trend relative to the cumulative degree hours on that plant response. So what we found was, it was quite interesting. This is, I would say this is the first time doing this level of study for watermelon. So it's probably, we should consider this is still preliminary finding, but it's very unique finding. So for the visual damage, it was more like this kind of response. This is the cumulative hours of chilling temperatures below 15 degrees C. So we also looked at different potential base temperature to consider the chilling. And then seems to be having a base temperature around 15 degrees C to show this cumulative influence, so that the bigger the number is, bigger the sort of visual quality damage. So you see that the threshold is suggested around this one. And then this is a visual damage, so it may not be so important. Most important one is this one. This is actually days from transplant to first female flower opening. And then it shows linear because we didn't have any data point here. So it could be more like, exponential type of response. But it shows that if you have temperature accumulated, then you have delay in terms of flower development, female flower. So that means yield is, harvesting timing is going to be delayed. And threshold temperature or the baseline temperature, I think, to consider the cumulative effect seems to be much lower than visual damage. And that was suggested around 4 degrees C. And then beyond 50 degree hours or so seems to be likely having some kind of significant increase in terms of flower development. So I thought this is quite unique finding we did for this out of this research topic. So I want to move on to the next topic. Strawberry production in greenhouse. So I'd like to give, again, a little bit of background. So North American strawberry production, it's very strong, but we have a seasonal increase or the seasonal up and down in terms of production capacity. California, this is showing monthly production and by region, and then blue and then dark blue are both California, Southern California and Central California. And then the green is Florida, and then Mexico is in red. So it shows, summertime we have a big capacity, mainly due to California capacity, and winter we have limited capacity, but Florida and Mexico are contributing. And then, in fact, they are competing each other too. Price point goes down during summer, goes up during off-season, wintertime and early spring. So there is a clearly opportunity for doing off-season production in US. Interestingly, this is something I saw recently. The whole production of US peaked at 2014, so the 2014, and then from that point declined sharply. I thought it's probably the market issue, but price is increasing continuously. So this is clearly showing the issue of producing strawberries in California, maybe because of the loss of fumigant, methyl bromide, maybe because of the labor cost increase, minimum wage increased, and then also exemption of the overtime changed. So those are actually hurting fresh vegetable and fruit production in California. And maybe also Mexico competing with Florida. So, of course, greenhouse growers know that opportunity, maybe supply chain knew that opportunity and pushed greenhouse more into the strawberry production. But now we have several greenhouse growers doing strawberry production in greenhouse, and then including California, Michigan, New York, Ohio, and Tennessee, in addition to Canada. And Mexico is already doing that too. Because of the cost of the production, typically they do a local production base and then targeting a little bit high price end rather than competing directly with Mexican strawberry or California strawberry. And then many of them are doing off season. So the summertime is usually not producing, but northern area like Canada, they can do year-round production. So it's very unique because not many countries can do year-round production of strawberry from the same greenhouse. This is one of the pictures I have. This is in fact in Belgium. It's beginning of December. Plants are already done with the first big crop production, fall crop production, and they are ready for going into winter break. And then after winter break, they start increasing temperature again. Winter break meaning lowering temperature, so the saving energy, and plants are going to rest during cold month of January, and then temperature goes back again and start producing in spring. So that's a sort of typical production cycle in Europe. And then that's the picture. But you can see that it's a window glass greenhouse. So typically you see tomato and cucumber and those crops now used in strawberry. All right, so when you want to introduce new crop in greenhouse or any sort of new system, you need to figure out a lot. So I had an advantage because I came from Japan, and then Japan has been using greenhouses for strawberry production, although their production is mostly on soil or soil bed based, but some growers are using hydroponics also. But still, we needed to figure out specific response using North American cultivars. So it was quite a bit of learning curve in my group. Initial 3 years, just trying to do something and then figure out it doesn't work and then figure out what is necessary research area. So we did that way. So starting around 2009, and we are still doing this. So it's one of the long-term projects we are doing. And of course, in Ohio, the focus is more for energy saving and things like that compared to what we did in Arizona. But one of the achievements we made in terms of introducing strawberry in the greenhouse was the finding of photoperiodic response. When you are dealing with field crop, you really cannot control light and temperature separately. But because of controlled environment, you can control light separately from temperature. That would create the necessity of finding temperature-specific response and photoperiodic response separately. But there was no information when we started what is the critical photoperiod of short day varieties of North American cultivars. Yeah, and then there are cultivars called day neutral, but they are not necessarily day neutral. So the first serious research or scientific experiment we did was finding out the photoperiodic response of selected cultivars or selected varieties of North American strawberries. So the left-hand side is showing a photoperiodic response of short day cultivars, and then showing that short day cultivars seems to have critical photoperiod between 13 hours and 14 hours. And then the right-hand side is the long day response, both ever-bearing or day neutral are basically facultative long day plants. So the longer the days, you can expect more advanced flower development. And then it was shown like that, 3 cultivars, Albion, Monterey, San Andreas, all responding positively to the long day. And then only 1 cultivar we tested showed almost the day neutral response. So not so responding to the day length. So that was quite important achievement we did, or contribution we did. Another one was a tip ban management of strawberry. So when we started doing strawberry production in Arizona, we had a massive incidence of tip ban, like 60% of the plant showing tip ban. And then tip ban is acceptable because it's a little bit of deformed leaves. So you can accept as long as the yield is good and plants are growing. But calyx ban is the issue. You can't market this brown calyx attached fruit. So we were basically committed to find a solution for this. Otherwise, we probably stopped doing strawberry and concluded, it's very difficult to grow strawberry in Arizona. But anyway, calcium deficiency is causing tip ban. And then strawberry should tip. You can actually see the risk or already the beginning of tip ban. It shows color, so the dark color. There is a mitigation issue available and mitigation measures available for other crops like lettuce. And I'm going to talk about that later. But strawberry, that measures cannot be applied because shoot tip is so deep in the canopy, so you can't really reach. So what we did was a different approach. So the guttation, we knew that guttation is a good sign of health. And then what we knew at the beginning, we never saw guttation in our greenhouse. So sure enough, we had a lot of tip-on. So the guttation happened when xylem pressure is maximized because of the root pressure. So that during the nighttime, usually in a coastal environment before sunrise, so that's end of the night, humidity is very high. And then so that completely shutting down the non-stomatal transpiration. So there's no way to lose water, but water is still coming from the roots, and then maximizing the xylem pressure and then squeezing xylem sap from the hydathode, which is basically opening at the end of the xylem. Then you see those water droplet. Whenever you see those droplet guttation, that means plants are happy, healthy, no tip-on. So what we did was creating basically coastal California environment in Arizona greenhouse at night. So increasing humidity to 95% to shut down these non-stomatal transpiration so that xylem pressure is high and then guttation happens. So it's proof of concept research. So the uncovering every day, actually every night, right? So the covering beginning of the night, uncovering beginning of the day, continue several weeks, and then we could see the big impact of doing that. So the tip-on reduced to around 10% from like 40%-ish. Calyx span also reduced to that level, so totally acceptable level. And then nighttime humidity was 95.3% inside this low cover, plastic cover. Without doing that, we had only 63%. So that's really the reason. But this is not feasible. If I recommend this to growers would say no way. So we needed to develop some more feasible technology. So we tested many things, and we ended up eventually with this underbench misting approach. Misting at night and then evaporating from the underbench, because we don't want to wet the plants. Wetting plants is the last thing we want to do, any crop production. So that would increase the humidity effectively. And then also, I think the one of the unique finding we did over the years of study was we only need several days a week to achieve that high humidity. And then each night, you only need 3 hours or so to achieve that high humidity. The rest of the time, you can reduce the humidity because it's really balance of calcium demand and supply. So if you can provide enough calcium to the growing shoot tip by doing high humidity treatment several days a week, few hours per night, that's enough. So that's actually helping whoever doing this, because humidification is, again, a big concern for inducing fungal disease, botrytis, particularly for strawberry. But great thing is that nighttime temperature for strawberry production is usually very low. So that's low side of botrytis activity. So 3 hours, several days a week doesn't really induce or cause a massive outbreak of botrytis. So we have been doing that every year in Arizona and also Ohio. Ohio, you may not know, but we get quite a low humidity in greenhouse too, because we are heating and the greenhouse is old, so the leaky. So therefore in that situation, you get quite a bit of reduction of humidity in Ohio greenhouse. All right, so my current graduate student is working on this comparison of cropping system, continuous winter production versus European standard double cropping, if this makes sense in doing Ohio or not. So we don't have results yet. So hopefully next time I talk, I can show cost analysis and stuff like that out of her research. So if you're interested in strawberry, that's a great news for us, because I hope more researchers start working on strawberry under controlled environment, because there are still tons of things we need to work on. So we are doing this year, actually coming up in 2 weeks, soilless strawberry school to basically teach the basics, mainly geared toward growers. But I think anyone who wants to learn about strawberry production methods and a little bit of science behind it is probably helpful. I have a website, so you can just go visit website and read and then find that's also effective, but this is another opportunity. So I just wanted to introduce that or advertise that to you. All right, so let's talk about different ones. So this is the leafy green research I'm doing in Ohio. And then one of the unique ones I'd like to introduce first is growing leafy greens under low pH nutrient solution. How low? I'm talking about like 4.0, 4.5. As you know, pH is a very important environment in the root zone, and you don't usually go below 5.5. You know, it's usually around 6 is optimum for most of the hydroponic crops. And then this is a classic study showing the pH response. And then you can see that pH 4 plants are showing significant reduction in terms of growth. pH 3, you don't even see the plant. So low pH doesn't work. We know that. That's why we are maintaining pH. But Oomycete pathogen, which is huge problem in liquid-based hydroponics, they like that pH range too. And then we know, as a common sense or common knowledge, reducing pH is one of the ways to mitigate the pathogen introduction, right? So the acidic environment are not good for pathogens either. So how can I, grow leafy greens under low pH? So why low pH is not good for plants? Well, so that pH itself, it is too acidic. Of course, it has a acidic, so that it has a direct effect. But certain range, maybe pH 4, 5 range, plants are not growing as good as pH 6 because their nutrient uptake is limited. Nutrient availability is different, or in some cases, like a substrate production situation, micronutrient is too high, and then just keep giving a toxicity. So some kind of nutrient related issue happens in that low pH side. So our thought was, okay, then what if we can just correct that and then adjust that so that plants can still take up nutrients and then maintain the growth under low pH? So we started working on and then discovered that pH response, or it's not probably discovered, it's confirmed that pH response is likely species specific. And then discovery was basil, interestingly tolerate all the way to pH 4. And then next slide is showing the growth, pH 4 all the way to conventional 5.5. The fresh weight didn't change, dry weight didn't change. Slightly reduced fresh mass of roots, but it was not affecting growth at all. Taste didn't change either. So I thought this is a great model to do the proof of concept study. So we did a challenge inoculation with PCM in the root zone, so the nutrient solution inoculated with zoospore or PCM collected from local greenhouse, hydroponic greenhouse grower site. And then you can see that root rot disease successfully introduced in a pH 5.5 nutrient solution on the basil plants. But under 4.0, you don't get that root rot disease. So the low pH prevented the infection, yet didn't reduce the growth. So again, we confirmed that growth was maintained at the same level as non-inoculated 5.5 pH. So that was quite interesting. But most of other crops, you get negative influence of lowering pH. So this is one of the sensitive ones. We tested so far spinach and basil and then lettuce. But we are testing more to see actual, range of the sensitivity of those very important leafy greens grown in hydroponics. But this is spinach, as I said, sensitive one. You can see the downhill reduction of fresh mass by reducing pH. And then this is a root morphology. You can see that a pH 4.0 plants are just not happy. But if you look at the nutrient uptake, it also, declining at low pH. So the first step toward improving growth under low pH was increasing nutrient concentration. So instead of, we could just work on those likely, limiting the growth, but instead of doing that, we decided to do just increasing everything so that it can be, same ratio. So we don't have to worry about antagonism of certain cations and stuff like that. So we tested low pH and high pH under with low EC and high EC. So the EC is the concentrations of nutrient solution under two different pH. So this is a conventional condition, spinach, 5.5 pH, and then about half strength of the nutrient solution, EC for 1.4. If we just reduce the pH to 4.5, you get less than half fresh rate. It's just confirmed with the previous, similar trend as previous finding. If EC is increased nearly 3 times, then you get recovery of fresh mass. So it's about middle point. It's still significantly reduced, so we need to probably further optimize the nutrient recipe for growing plants in similar or comparable level with the conventional. But it's a good sign. And then actually for this lettuce experiment, this is only this one particular one-time experiment, so we need to repeat that. But the low pH, high EC actually brought the fresh mass almost the same level as conventional conditions. So that was quite promising. And then again, as I said, we have a new graduate student and working in this technology development, testing many different species. And then we will be eventually developing nutrient formula that we can suggest for growing leafy greens under low pH. So the last part, using a few more minutes, I like to talk about is a tip pan management, which has been an issue in controlled environment and still an issue in controlled environment, and more issue or more problematic issue in indoor controlled environment like a vertical farm. So just recap, tip pan is a calcium supply-demand imbalance. It's a localized imbalance of calcium between supply and demand. Demand is coming from growth, and supply is coming or driven by mass flow, in this case, transpiration, or root xylem pressure, like I show in guttation slide. And so we know that transpiration is very important for tip pan. Whenever you enhance transpiration, you have a mitigation of tip pan. And then particularly internal part of the plant is very important. So you want to enhance the internal or young leaves transpiration rather than old leaves transpiration. So understanding transpiration, right, in tip pan is very important. So to apply this type of setting, however, there is a huge limitation. Well, so let's look at the methods of transpiration measurement. Direct measurement by water uptake, yes. If it is a simple system, only one stage of plants are growing, in that system, you can just measure the water consumption and then find the transpiration. That's fine. But most of the cases, mixed cultivars, species, and mixed stages. So it's really, not possible. Direct measurement of transpiration, there are scientific equipments available, leaf gas exchange system or sap flow gauge. That's possible, but not applicable in commercial setting or leafy greens. Engineers love to put a lot of sensors, net radiation sensor, airflow, BPD sensor, temperature sensor, and then try to find the aerodynamic method to find the transpiration, all that. But it's such a teeny tiny headspace. It's no way to do that. And then also very expensive to do that. So we wanted to develop something very simple growers can use. So we decided to use this simple Petri dish approach. So the if you are familiar with evaporation, evapotranspiration measurement in open field, you probably have heard of pan evaporation, which is basically big dish, like a 1.5 meter diameter, but we are doing micro scale measurement, similar system using Petri dish. But we had to change the color of water. Unlike outdoor, our light source only emit the sort of visible wavelengths rather than, the entire shortwave wavelengths. So, and then water doesn't absorb really well. Absorb the visible light very well. So you have to change the color of the solution. But with that, we found that it can be a really good tool to actually assess the microclimate in that small, teeny tiny petri dish, and then spread those many, petri dish in a growing environment so that we can map the potential transpiration in that growing space. And then, by the way, the master's student who got a bachelor degree in engineering is now working with me for master degree, and he did all this study, geo. So we found that it's a quite useful. I show you a few more examples. So this is a walk-in growth chamber, and then he had two spaces for measuring, and he put the petri dish, and then also another shape of container for detecting the influence of direction of airflow. But anyway, he could map the potential evaporation or transpiration at different conditions, like a low airflow, high airflow, BPD, different BPD, highlight very nicely. And then he could see that correlation. And then we are validating the response of this simple sensor compared with more advanced way to quantify transpiration. So we think this is going to be quite useful tool for growers to assess the potential transpiration, and then they can compare with growth side, and then they can assess the risk of tip ban. So in that application or area of study, indoor farming, I'm also doing a lot of outreach type of activities. One of them is Indoor Ag Science Cafe. It's a monthly forum for growers or ventures who wanted to do indoor farming. So basically selected topics presenting, and then the open discussion. So if you're interested, please go to this website, and then there's a sign up form, so you can sign up, or just email me, and I'm happy to include you, anyone who is interested in that mailing list. So Ohio State is investing more for the controlled environment agriculture also, just like you're doing for in Texas A&M. Building a new greenhouses in Columbus campus. And then also we just formed the Center for Controlled Environment Agriculture with 18 faculty members working in controlled environment, in addition to other areas too. But my focus, and then some other people's focus, have been controlled environment agriculture. So with that, I would like to thank everyone who was with me today. And then I'm happy to take any questions.
Thank you, Thierry. It was great seminar. So you provided so many nice tips, from the different 3 or 4 model crops that you use. So that's great. So the floor is open for questions. I don't know if there is any in the chat.
There's nothing in the chat thus far, Daniel.
Okay. Well, I guess I start, Thierry. So just the first part, you talk about the problem in tomato, the rootstock with the intumescence, and then you show that that was no cultivar specific, and I believe the cultivar that you have that was Beaufort.
Mm-hmm.
Have you screened also? I'm sure you screened some other rootstock genotypes that may not be genetically related maybe from the seed company, or what have you seen with that respect? If you use, I don't histamine or DRX?
No, we haven't. That's a probably important, to do. So we know some scion varieties are susceptible or sensitive, and then Beaufort and all that derider varieties of rootstock interspecific are very sensitive to this. But we never had the opportunity to screen or test or phenotyping, including other seed companies' rootstocks.
Okay, yeah. All right. And
yeah, so that's one of the great questions. One of the typical questions I get from growers, and then I don't have a data supported answer yet. 1 year probably easily. Beyond that may not be so easy. So the continuous production I'm talking about, but I know it's not a day neutral based study, but indoor grower in Japan doing strawberry production in totally enclosed, controlled environment with fluorescent lamps. I think they're using. They go up to 2 years. Yeah, plants are looking quite unique. I have seen picture. It's all stretched because so many leaves. Just was right. Yeah. So I don't have a definite answer, but I have seen more than 1 year grown. Whether it makes sense or not, I think depending on market.
all right. So there is another question in the chat, Cherry, and that relates to the problem again on intumescent. So if you grow plants under LED without UVB, is there any treatment other than changing the light quality to mitigate this intumescent problem? And the second part of the question is if you know either the physiological or biochemical mechanism of intumescent, if it has been discovered or investigated further.
Yeah, so the first question was, actually I can scroll up and see. So mitigate intumescence. So I think many people worked on humidity as one of the factors affecting intumescence. And then humidity is, yes, to me, it seems like humidity is sort of, already intumescence is initiated and then high humidity condition and then exaggerate the situation. So that increase the severity of intumescence. So that's humidity influence. Other than that, we didn't, light quality is the one. Other than that, we didn't have any knowledge about any other factors. Or physiological mechanism. Yeah, so it's a good question. So I think it's a water relation related, and then how UVB is preventing intumescence. I think most likely just making the epidermal layer harder. Or it's not really scientific terminology, but preventing plant, to get swollen cell or too much swollen condition. So the UVB and then also blue light, I think, are affecting that way to suppress. Far red, I think it's just helping to stretch the cell to a longitudinal way or the opposite way relative to the swollen direction. That's probably the only way I can explain why those factors helping. How it is induced, it's a great question, but I don't have really physiological good explanation for that.
Right. You provided some ideas. Thank you. The next question is in relation back again to the controlling tip burn using high humidity. So can this strategy of high humidity be used to increase nutrient delivery, other nutrients more in general speaking, beyond the tip burn?
Yeah, so anything mass flow driven, so the calcium, magnesium likely enhanced by a high humidity guttation approach. So if you have a magnesium deficiency, for example, and then can't solve that issue otherwise, then high humidity probably helps.
Okay, thank you. Another question is, how could supplemental UVA or UVB light affect tomato plants from the pathology perspective?
Good question. So the UVB, I think, was studied by pathologists in terms of pathogen infection. And then I think it should work. I think strawberry is one. I think it's a UVC, not UVB, but I think there is a positive response reported. UVA, probably not. UVA is more like a blue, so not at the same level as UVB. You probably need to apply a lot of UVA to get a similar response.
Okay. Another question from Sam. A follow-up from Sammy. How about UV light affecting microbial mobility from the medium?
Or in the hydroponics? Yes. So UV is used for disinfestation of nutrient solution, but you have to separate UV light from growing environment or the root zone of where the roots are, because you don't want to damage the roots by UV radiation.
All right. Sounds good. So I think it's Trish, you're keeping track of the timing. But,
yeah, it's a couple of minutes after 5. So I think if anybody has, is there one more question that anybody would want to ask? You're welcome to ask it.
I have one final, maybe following what Dan, it was related to the photoperiod in the strawberry. Can different temperatures affect the optimum for photoperiod for short day versus day neutral?
Yeah, so that's an interesting area. I wish to do more research in that, but strawberries are very unique. So the short day plants, once flowers are induced under short day, as long as temperature is in a certain range, you can even apply long day. There's an interaction between temperature and short day. And then also induction of flowers can be done with just low temperature also. Another factor affecting flowering induction is nitrogen fertilization. So all three used by growers to induce flowers. Otherwise, you can't do under like a long day condition. Day neutral, also similar, low temperature also kind of enhance the flowering, but day neutral or the ever-bearing long day type. Controlling flowering is more challenging in that because they are facultative long day, so that means they flower anyway, and then switching on and off is more challenging for that type of varieties. It's a lot of interesting areas we need to work on.
Yeah, it's very interesting. I remember when I was a undergraduate student back in Argentina, part of what I was doing is producing some strawberries just to get some money to pay for the studies. And one way to get into the market early, so we have a section. So I put the strawberries next to trees, like poplar trees, the curtains. So you have a little more shadow to reduce the temperature. That allows about 7 to 14 days early flowering and entering into the market at a high price. So this is only on the short day types.
Mm-hmm. Yeah, interesting.
Yeah.
Yeah.
Okay, we have one more question in the chat. This is it. Is there any relation between light and tomato fruit firmness?
Oh, tomato fruit firmness. So the tomato firmness can be firmer, I would say, when you have low humidity is one thing, or limited transpiration by high EC. So those are so that water relation. Light, probably light intensity, because it would increase the transpiration, but not light quality. I don't think light quality would affect firmness.
Okay, well, thank you. Well, let's all thank Dr. Kubota again for an excellent seminar. We really appreciate you taking the time to be with us this afternoon. And our next seminar in 2 weeks will be Dr. Ed Harwood, who is the Chief Science Officer for Aero Farms out of Newark, New Jersey. So Sherry, I hope you can join us for that. If you have time, we'd love to have you continue to join us for the rest of our seminar series.
That's wonderful. Thank you so much.
Thank you. And we'll see everybody in 2 weeks. Bye-bye.
Thank you. Thank you, Cherry.
Thank you, Daniel.